Welcome and thank you for attending today's webinar, Beta Cell Screening in Vitro Electrophysiology for Diabetes Research. My name is John Batista and I will be the moderator for today's presentation. Beta Screen is a novel suite of tools including multi-electrode arrays, fluidics control, and software designed to facilitate the analysis of electrogenic activity of the islets of Langerhans in both acute and chronic settings. Today's presentation will be delivered by Dr. Sven Schönecke, who is joining us from Multi-Channel Systems in Reutlingen, Germany. Just some notes before we begin. First, the webinar is being recorded, and after the event, everyone will receive a link via email to that recording. Second, if you'd like to ask a question during the event, feel free to type your question into the Q&A module at the bottom of the presentation window. Please note, this menu auto-hides and may require you to mouse over to see it. At the end of the presentation, we will have a question and answer period where we will try to answer as many of your questions as time allows. If we miss any questions, they will be answered directly via email. Please allow me to introduce today's speaker, Dr. Sven Schönecke. Sven is a senior application specialist at MCS, where he has worked for the past 3 years. He received his PhD in biology from Eberhard Karls University in Tübingen. I will now turn the floor over to Dr. Schönecke.
So, hello everybody from my side as well. And John, thank you very much for your kind introduction to our webinar today. And as already mentioned, we'll talk about beta cell screening using microelectrode arrays, in short MEAs. And today I will mainly talk about acute and chronic recordings of electrical activity in intact islets. And the idea is that I will guide you chronologically through the recordings I did in the first years of the project, and I will show you the progress we made with regard to the recording technique, the MEAs, and the hardware. Let me introduce you shortly to the islets of Langerhans. Of course, they are part of the pancreas. And the pancreas has two main functions. So the exocrine function is to digest, to secrete digestive enzymes into the intestines, and the endocrine function is to regulate the blood glucose concentration. And this is done by the islets of Langerhans, and the islets are distributed over the complete pancreas, with the highest density in the tail of the pancreas. And the most important cell types are alpha cells, beta cells, and delta cells. And if we go down to the cellular level, the beta cell is the only cell type in the body who can produce and secrete insulin. And insulin is the only hormone who can effectively lower blood glucose concentration, and therefore is essential for survival. And the mechanism of glucose-induced insulin secretion is also called stimulus secretion coupling. Let me show you briefly the stimulus secretion coupling. So postprandial, after food intake, the blood glucose concentration increases and the glucose gets into the beta cells via glucose transporters. Inside the cell gets metabolized via the glycolysis, and energy-rich substrates are transported into the mitochondria where ATP is produced. And in the close vicinity of the KTP channels, the ATP-ADP ratio increases. This leads to an closure of the KTP channels and is followed by a depolarization of the cell membrane. This, on the other hand, activates voltage-gated ion channels, mainly L-type calcium channels, and this leads to an influx of ions into the cells. And this is the direct trigger for the exocytosis of insulin. This event, of course, is very important and recordable. But the beta cell is much more than only calcium action potentials. So I want to show you these two figures from two important papers from Santos et al. And Guillon et al. And they showed for the first time that everything which has to do with the stimulus secretion coupling oscillates. So in the upper trace, you can nicely see the intercellular calcium concentration inside a beta cell, and simultaneously recorded the membrane potential, which also oscillates. In the lower part, you can see the green trace, which is again intercellular calcium concentration, and with a short delay in red, the insulin secretion. So oscillations are very important, and I want to show you this slide here where you can see electrophysiological recordings with intracellular electrodes from my former working group, and they show activity in different glucose concentrations and 7, 10, 15 and 20 millimolar glucose. And you can nicely see oscillatory activity in these cells. I show you this slide because everybody who ever did such recordings knows how difficult it is to record with intracellular electrodes. So one recording a day is already a good result. And therefore, I would like to compare the classical electrophysiological methods with the new MEA recording methods. Because, of course, the question arises, why should we use the MEA technology for such recordings? And the reason is that classical methods like the patch clamp technology or recordings with intracellular electrodes have disadvantages. So first of all, you need high technical skills to do such recordings. All of these recordings are invasive, and this leads to that the long-term studies are impossible, and all of the recordings are endpoint recordings. So, and at the end, this leads to only low throughput recordings. And the MEA method has the advantage that you record from a planar electrode on a substrate. And what you do is you record extracellular field potentials. And the advantages here are that the recordings are non-invasive and you can record from intact islets. It provides you with some simple experimental handling. It provides you with a higher throughput, and also long-term recordings are possible. So these are the, I think, most important differences between those techniques. And a few words to the microelectrode array technology, in short, MEA. They are produced here in Germany in a clean room, and a standard MEA looks as follows. So you have a glass substrate, contact pads, a culture chamber on top of the MEA, and an electrode array with the recording electrodes in the middle of the chip. There are various types of different MEAs available. Here are examples of a 6-well MEA and a multi-well plate for higher throughput recording. So it depends, of course, on the application and on the customer wish what you want to use. A few more words about the MEAs. So a standard MEA has 60 electrodes, and the electrodes are made mostly of titanium nitride, but also gold electrodes are available. And the customer can decide what kind of electrode size you want to use, what kind of inter-electrode distance, and also the amount of electrodes. And historically, the MEAs are used mostly in the neuroscience field or in the cardiac electrophysiology field. And you can see some examples here on these photos, for example, a brain slice. Let's come back to the islet recordings. So I would like to introduce you to the establishment of the MEA technology in the field of experimental diabetes research. And I want to show you how extracellular electrophysiological recordings with intact islets on MEAs look like. And I want to show you how we validated the new recording method, of course, with modulations of important ion channels. So at the very beginning of the project, I started with some standard MEA, so you can see here, and I used mouse islets. And I simply pipetted a mouse islet into the array field and placed or positioned the islet with the help of a micromanipulator and a glass pipette on top of the electrode. It is important to have a good contact to the electrodes to get a good signal-to-noise ratio. And a standard recording looks like this, so from left to right. And during the application of 3 millimolar glucose, which is under the threshold of activity, at least in mouse islets, there's no activity detectable, only a baseline. After the increase to 10 millimolar glucose, you see an initial strong burst of electrical activity, followed by a short break, and then the characteristic oscillatory activity takes place, which is stable over a very long time. Interestingly, there are two phases of insulin secretion, the first phase and the second phase. And you can nicely see those two phases on the MEA recordings. The first phase is the first burst, and then the second phase are the oscillations. You can nicely see on this slide the proportionality of glucose concentration and electrical activity. Also, again, from left to right, I increased the glucose concentration from 3, 8, 10 to 50 millimolar. And you can nicely see that the higher the glucose concentration is, of course, the higher is the electrical activity. And this matter of fact helped us to make a quantification of the glucose-induced electrical activity. And we used mostly the fraction of plateau phase in a short FOB, which is shown here on this drawing. So a synonym for the fraction of plateau phase is the percentage of activity. And you can see here on the right side our first dose response curve. The FOB plotted against the glucose concentration. And important is here the effective concentration, the EC50 value, which is around 12 millimolar. And this was absolutely in line with recordings with traditional methods like intracellular electrodes. And it was the first hint that this technology is more than an alternative. So these data were published in our first MEA publication. Further, we did validation studies of the new recording method. And in A, you see the modulation of the KTP channel. So I used diazoxide, which is a strong KTP channel opener, and tolbutamide is a strong KTP channel blocker. And you can see that during the application of diazoxide, the electrical activity was strongly inhibited. And after I added tolbutamide in a higher concentration, it restored electrical activity. A similar experiment is in, you can see in B, where I applied nifedipine as an L-type calcium channel blocker. And you see that nifedipine inhibited electrical activity, and the effect was reversible after the washout. Oscillatory activity slowly came back. You can see nicely in the end of the trace here. On the right side, I did a recording where we recorded simultaneously electrical activity and intracellular calcium concentration. So the black trace represents electrical activity, and the red trace, the intracellular calcium concentration. Note that each burst of electrical activity is accompanied by a peak of calcium. And this is also part in our, also published in our first MEA publication. So further on in the second part of the talk, I would like to talk about the effects of oxidative stress on the electrical activity of islets of Langerhans. And the reason is that is the development of type 2 diabetes mellitus. So first of all, there are two main factors for the development of the disease. First of all, there's a strong genetic predisposition, of course, but even more important, a very strong correlation to modern lifestyle. So that means chronically increased glucose and lipid concentrations leads to overweight. And these stress factors were generated then in form of oxidative stress or reactive oxygen species can directly affect beta cell function. And the question was here, are stress-induced effects detectable via the MEA technology? So I induced oxidative stress via hydrogen peroxide, H2O2. And you can see on this example recording that the effect was directly detectable with the MEA system. And also the quantification in this dose response curve here, the FOB plotted against the H2O2 concentration. So this was a nice start for this experiment. And then the next questions were, where are there strategies to protect the cells against induced stress? And even more important for me in those days is the resolution of the MEA technology high enough to detect such small effects? So to protect the cells, we used an antioxidant named Tempol. And what I did is I incubated the beta cells or the islets in Tempol. And what you can see here in A is, first of all, a control recording with normal activity. Then a recording where H2O2 induced a reduction of electrical activity. And then I did the same experiments again with islets pre-incubated in Tempol. And you see nicely that there was a protective effect. And in B, you see just the quantification of the fraction of active phases. And we did a further experiment where we checked the effect on insulin secretion, and we could see similar results. So the take-home message here is MEAs can even detect small changes in electrical activity of islets, which makes it to a very good tool for pharmacological recordings. Next important milestone was the establishment of long-term cultures of intact islets on MEAs. And the reason or reasons for that was that classical electrophysiological methods are, as I mentioned already, are invasive and only suitable for endpoint recordings. And it is a fact that there is no established electrophysiological method which enables in vitro long-term studies. And again, back to type 2 diabetes, it is a disease which takes quite a long time, a long-lasting effects. So I think it is very important to have an in vitro model where you can study stress factors and substances against these stress factors. So the further advantage of the islet cultures, of course, is that I can record from several islets per experiment, and I can do recordings over a long time. And that this works, you can see here. So there are two example recordings in A. The first recording was after 6 days in culture, and you can nicely see that the islet responded to 10 and 50 millimolar glucose nicely. And then the second recording on the right side, you see a recording after 34 days in culture. And you see that the islet still responded to 10 and 50 millimolar glucose. And after the quantification of the data, which you can see here in B, it doesn't matter if you look on day 6 or day 7 or on day 33 or 34. So the electrical activity is remains stable over a very long time. And even more important, we could directly compare the cultured, from the recordings from cultured islets with the acute recordings we did before. So that the take-home message here is electrical activity of islets is preserved in culture over a very long time on top of microelectrode arrays. The data are also published in our second MEA publication. And the next part of the talk, I will discuss the MEA 2100 beta screen system. And the aim was here to develop a parallelized screening device for acute recordings. So to obtain a higher throughput recordings, which are also suitable for drug development, but also suitable for basic research in the field of diabetes mellitus. And it could be also used in the future as a quality control system prior to islet cell transplantation, for example. So back to the beginning of my talk, we placed the islets for acute recordings with mechanical pressure from above on top of the electrodes. And the idea was with the beta screen to somehow position the islet via a negative pressure on top of the electrodes. And we could see that this is the way to go. As you can see on this photo, you could nicely place islets on top of the electrodes via negative pressure with the help of a vacuum pump. And you see here already in A, 3 electrodes placed on a polyimide foil and surrounded with positioning holes here in B and C. And in photo D, you can see a complete recording chamber with 5 electrodes and nicely positioned islets on each electrode. And I did that with 10 millibar negative pressure. And on the lower right side, you see an example recording in 10 and 50 millimolar glucose. And what we realized after the first recordings is that the quality of the recordings was even improved in comparison to the former recording technique, because we had a better contact to the electrodes, and the signal to noise ratio was much better, which means the signal amplitudes were increased. Of course, we did also validation studies with the beta screen system, and we found out that the EC50 value is absolutely comparable to our former recordings and also to literature. And also the modulation of ion channels worked as expected, so that we can say that electrical activity recorded with the beta screen system is in line with literature and increases the throughput by a factor of 40. I show you why and how. But before, I will also talk in my last section about the establishment of human islets recording on MEAs. And of course, there's an increasing interest in human islet recordings, because in the last 5 or 10 years, scientists found more and more differences between human islets and the animal models. So this is an important point. And another important point is that of course there's limited access to human islet biopsies. So it is important that the few islets you have, that you can record them as quickly as possible, which is impossible with traditional methods. And of course, it could be also used as a quality control prior to islet cell transplantation. So we tried, so these photos are actually made before the beta screen device was developed. So I tried different techniques. So I placed the islets via mechanical pressure. I did pancreatic slices, you can see here in B. I tried to cultivate the islets on top of the MEAs. And my very first recording looked like this, which is also published in our third publication. And you see that we could see activity in 10 millimolar glucose, and then I realized, okay, I will probably lose the activity, so I added additionally a little bit of tolbutamide, and I could restore electrical activity. So to my knowledge, this was the first recording of a human islet on top of MEAs. I want to show you this slide here because it nicely shows the advantages of the MEA technology. So you can easily compare different species. You can easily compare wild-type mice against knockout mice, for example. I did just two simple recordings here. So I used a human islet and I added tetrodotoxin, which is a very strong sodium channel blocker. And you see a strong effect on the electrical activity. And on the other hand, I did the same with a mouse islet, and there was absolutely no effect, which nicely shows that they are differently equipped and the stimulus secretion coupling works differently in different species. At the end, a recording from a human islet on top of the beta screen system. So I applied different substances and tried different things. But I show you this trace here because you see that here's activity. You can also inhibit the activity, but you don't see these oscillatory activity here. So that was what I saw quite often with human islets, that I saw more single spike activity than these clear oscillations like on mouse islets. I don't know exactly why, but I found out that also Rossman et al., for example, published a paper where he described similar things. He did recordings with patch clamp, and he also found out that there's more single spike activity in human islets, interestingly. So here I want to show you the increase of throughput. So this is a single recording with a beta screen device. So I recorded simultaneously from 10 different islets with a beta screen, and each row represents a recording. And I just show you that because I did this recording within approximately 60 minutes, and I had a complete dose response curve. And before, with at the very beginning of my project, this took me maybe 3 or 4 days to get such many traces. So this is really an improvement. Let me talk about the available devices we have at the moment for chronic and acute recordings. So for cultured islets, for chronic recordings, I would recommend the incubator ready system, the so-called MEA 2100 mini system. The big advantage of this system is that you can place the complete head stage inside a wet incubator. So you don't have to move the system. You can just put the MEA with your cultured islets into the system, and you can record under stable conditions for a very long time. If you are more interested in higher throughput recordings and drug testing or drug screening, I would go into the beta screen direction. And I want to show you how the beta screen looks in more detail. So you see a beta screen head stage here, and then a specialized MEA for the beta screen system. The MEA is equipped with a perfusion inflow and outflow. You have a connection for the negative pressure, and of course your MEA with 5 electrodes. And you can easily upgrade the system to 8 head stages, and because of that you can record from up to 40 islets simultaneously. And of course, we have also a specialized software for the beta cell recordings called the Beta Screen Experimenter. It is the software is specifically designed for long-term and acute beta cell recordings, and has a specialized tool set for oscillatory activity and spike analysis, as you can see on this example. And you have an online analysis of those response curves. So after you press the stop button, you have already your results. So I think I want to make a summary of the talk. So I hope I could convince you that the MEA technology allows electrophysiological recordings from intact islets, provides a simpler experimental handling and higher throughput than traditional methods. It meets the needs of academic and industrial laboratories. And extracellular recordings enable long-term in vitro research and co-cultures. And it is suitable for drug development. And also recordings with human or stem cell derived islets are possible. Thank you very much.
Thank you, Sven, for a very informative presentation. We're going to be taking questions that you may have. If you'd like to type them into the chat window, that would be helpful. As one of the attendees has already done. So the first question is, does the negative pressure used with the beta screen have any effect on islet cell activity?
So what we, yeah, it's a good question. So as I showed during the talk, we did those response curves, and what we saw is that there was no effect of the negative pressure to the recordings, as the EC50 value was absolutely comparable to the other EC50 values.
Okay, thank you. And another question just now, the human cells used for the recordings, were they from a healthy person or a type 2 diabetes patient?
So the human biopsies were from non-diabetic patients exclusively, always.
Okay. And a third question, what is the longest period established that islet cultures have survived on MEAs?
Okay, so what we published was until day 34 in vitro, but I did recordings up to 65 days. But I realized that after a certain period of time, so it's difficult to say, but I would say after maybe 50 or 60 days, I could observe that the activity decreased. But we could see reproducible activity until day 34.
Okay. And another question. Can you record from alpha cells on MEAs?
I only recorded from intact islets, and I never saw alpha cell activity. But to be honest, I'm not really searched for alpha cell activity. But there's a group in France, in Bordeaux, and they published also alpha cell activity on MEAs. But if I remember correctly, they dispersed the cells. So they had not intact islets anymore, so cell clusters, and probably a cluster with many alpha cells on top of the electrode. So in theory, it's possible, but I never recorded alpha cell activity by myself.
Okay. And finally, are co-cultures possible on top of MEAs?
Yes, indeed, it's possible. So this is also done with neurons or cardiomyocytes. So on top of an MEA is quite a lot of space, and you can also add membranes and different things into the MEA. So of course it's possible. And I think this is a really interesting and promising application for the future.
Okay, great. Well, that seems to wrap it up. You can see the slide on your screen that contains our contact details arranged by geography. Please take note of those email addresses and phone numbers. And if there are any questions that arise at the conclusion of this, feel free to send us an email, and we'll do our best to respond in a timely manner. So with that, I want to thank again Sven for taking the time to take us through this very informative presentation, and all the attendees for taking time out of their day to be with us.
Thank you very much.
Okay, this concludes our presentation. Have a good day.
You too.
Bye-bye.
Bye-bye.